S in Microbiology. I have cleaned my station as well as placed on my PPE. I've also prepared our lysolized E. coli culture, M. luteus culture, and E. epidermidis culture. Now the first thing we want to do is grab our E. coli, make sure it's nice and mixed up, place that back on the rack, grab one of our inoculating loops, pull that out, grab our E. coli, flame the mouth, and then we're going to aseptically grab a sample, flame the mouth again, place that cap back on. I then have my triple sugar iron agar labeled with E. coli at the top. I'm going to flame the mouth of this one. And then for this, I'm going to enter the loop all the way down, pushing into the bottom, coming back up, and moving zigzag all the way up to the top. I'm now done with that loop. I can reflame this mouth. I will place back on the lid, slightly open, and place that back in the rack. Next, take my M. luteus relysolized culture, grab another inoculating loop, close that back up, take the cap off, flame the mouth, grab a sample, flame the mouth again, and then close that up. I will then grab my M. luteus TSI agar, open the lid for that, flame the mouth, stick that all the way down into the agar, and then come back up and do a zigzag all the way up to the top. We're now done with that inoculating loop. Reflame the mouth, place that back on, turn it a quarter back. Last but not least, I grab my S. epidermidis, Mix that up. I will need one more inoculating loop. So I will pull that from the bag, close that, open up my S epidermidis, flame the mouth, get a sample, flame the mouth again, and replace the cap. I will then grab the TSI agar labeled S epidermidis at the top. Flame the mouth. Enter this into the tube, breaking the agar, then comes back up, and I do a zigzag all the way to the top. And then flame the mouth. And place the lid back on, turning the lid slightly